Assalamu alaikum. Respected chairs, moderators, and esteemed audience. I am at the very last version of this presentation, and you all are practicing oncologist. I wonder that you might have the most boring 10 minutes. So my topic is microRNA 27A deregulates human heparin-binding EGF-like growth factor in ovarian carcinogenesis. So before in the evening, I was asking ChatGPT what might be the future direction of research with microRNA before going to the topics. So I found microRNA hold great promise as therapeutic agents in cancer due to their ability to regulate multiple oncogenes and tumor suppressor pathway simultaneously. This is important point. Another point is microRNA can restore and modulate cancer progression, enhance treatment response, and achieve more targeted and personalized therapy. So with this objective, I am going to introduce you about microRNA. So this is the short non-coding RNA, and they have some binding site in the 3' messenger RNA tail site, and they have target for all genes in 60% genes. And microRNA biogenesis like that, it is synthesized in the nucleus, they come out and they finally bind here, they refresh two stage, translational repression and micro-mRNA cleavage. So microRNA works in two stages, mRNA degradation and translational repression. So this is cancer. In cancer, we find whatever the phenotype is, there should be some genotypic sense. So genotyping says this is normal cells, DNA repair genes are downregulated, tumor suppressor genes are downregulated, oncogenes are upregulated. So all three things will happen, or part of these three things will happen. And finally, we will get malignant cells. My question is, what about microRNA in the cells? So if microRNA is upregulated in tumor cells, microRNA upregulation occurs and tumor suppressor gene downregulation occurs. And in tumor cells, if microRNA is downregulated, the oncogene expression will be promoted. And finally, in ovarian cancer, this is the most lethal of the human female cancers, which there is no reliable screening test. And hHB-EGF, this is a human heparin-binding EGF-like growth factor. This is a gene that is responsible. This is an oncogene. It is responsible for oncogenesis in ovarian cancer, and this is the biology of this gene. I will skip this one. And our hypothesis is microRNA might play potential role in ovarian carcinogenesis by targeting hHB-EGF gene and modulating its expression. So our objective is like that, to find out the candidate notorious microRNA that binds with hHB-EGF and investigate the functional role of candidate microRNA that deregulates hHB-EGF in ovarian carcinogenesis and find out the therapeutic potential of the candidate microRNA. For that one, we use target scan and mirror-based software. We found a pool of microRNA. From that microRNA, we could carry out the screening. After screening, we get candidate microRNA and then carried out functional analysis. So we get 11 microRNA that were supposed to bind with target hHB-EGF, this gene. Among these 11 microRNA, then we introduced ovarian cancer cells. These 3 ovarian cancer cells, this one SCOP3 MCAS. These 3 ovarian cancer cells have differential expression of hHB-EGF in 3 types. Then we carry out by quantitative real-time PCR, we check the expression of our targeted microRNA, and we found that in this microRNA 27A, 27B, and microRNA 99A, this has reciprocal relationship with hHB-EGF gene. And then from there, we screened out microRNA 27A because it has reciprocal expression with hHB-EGF in this part. Then we targeted microRNA 27A as a more promising candidate for our hHB-EGF down regulation and carcinogenesis. And here is then we check that whether this microRNA can target hHB-EGF by carrying out luciferase reporter assay. We check that this microRNA here, this MIR27A targets this hHB-EGF in that 5' RUTR. And then we carried out check that microRNA expression, how it modulates the hHB-EGF expression in the breast ovarian cancer cells. We found microRNA 27A deregulates hHB-EGF by 50%. And in MCAS cells, in another type cell, this regulation was less. So we checked from here, we selected microRNA 27A and scope 3 cells, and then we check that whether the mechanism by which microRNA 27A modulate hHB-EGF in scope 3 or ovarian cancer cells. Here we found that there is down regulation occurs like that in mRNA level by real-time PCR, and in Western blot analysis showed in protein level. So we could conclusion that microRNA binds deregulate ovarian hHB-EGF in ovarian cancer cells at protein level and mRNA level. Then we carried out microRNA, how it affect the in vitro growth of ovarian cancer cells. In vitro growth, 3D collagen assay and soft agar colony formation assay. In both cases, we found that microRNA, this 27A down regulates hHB-EGF cell number. So mRNA expression, if we increase, and that cell growth was decreased. By that, then we introduce this cell to check the in vivo assay, in vivo tumorigenic assay in biopsy node mice. Biopsy node mice is immunocompromised mice. Then from this here 1 million cells we are inoculated in this node mice, and after 7 weeks we found this type of here non-control, we found big tumor. Here also anti-microRNA big tumor, but this was microRNA overexpressing cells, and this tumor size was significantly less. We separated the tumors, we found like that. And these data we are then, we carried out dose dependence, not dose dependent, in the timeline. And timeline from we found that from this non-control and anti-mir compared to control group, microRNA 27A induced case, the tumor cell were suppressed and tumor growth was inhibited in this mice. So this gave us that microRNA 27A in vivo condition, it has the potential to inhibit the cellular growth of these ovarian cancer cells. So from here, actually after that, with my collaborator in Japan, we are also checking the phase 1 trial, but I am showing you the data of up to this part here. And then from here the take-home message is microRNA 27A acts as a tumor suppressor in ovarian cancer by directly targeting and down-regulating hHB-EGF, a key molecule that is involved in cancer cell proliferation and tumorigenesis. This finding highlights the therapeutic potential of modulating the microRNA 27A and hHB-EGF axis as a novel strategy for ovarian cancer treatment. And microRNA, not only this microRNA, has the potential to be used as biomarker. And I hope, I expect we will show you some data in the next year, 2026, about the use of microRNA as a biomarker of cancer, especially for some sorts of cancer detection. Thank you very much. And I would like to acknowledge TBRF, Osaka University, Khulna University. Thank you. And finally, let's fight together, and we want to fight our male and female all cancers together. Let's stand together against cancer. Thank you so much.